Okay, so in the previous video, we learned about codons and how each codon codes for an amino acid. Now we're going to talk about how does the amino acid arrive to the ribosome. So we have these special molecules called tRNA's. That stands for transfer RNA. Now a transfer RNA is a single strand of RNA that folds up. Now at the bottom of the transfer RNA, the tRNA, are 3 nitrogen bases, because the whole thing is made of nucleotides and nitrogen bases, but it just folds up on itself. And these 3 bottom ones are called the anticodon, which is what I'll talk about in a little bit detail on the next slide. But here at the top is where we have the amino acid attached at the top of the tRNA. So this here is the anticodon. So let's go ahead and check out tRNA's in a little bit more detail. So you can see here how it's a single strand, a single RNA molecule that kind of folds up on itself. We can see how it hydrogen bonds or base pairs together right here. Now at the very bottom of this tRNA molecule, we have the anticodon. Now this anticodon is really, its main purpose is to attach or bond with the codon. Now, if you remember, the codon is what codes for an amino acid. So this codon, UCA, is what you would use when using like a genetic code. Oops, sorry. Can you see it? There we go. When using the genetic code, you would use a little table. You'd find UCA. You're like, oh, that's serine. Well, now to bring serine, we're going to bring serine using a transfer RNA. So if you notice that anticodon base pairs with the codon. So if there's an A in the codon, an U in the codon, there's an A in the anticodon. A C in the codon, there's a G in the anticodon. An A in the codon, a U in the anticodon. I also want to just point out for some classes that third base, in this case it's the A to U, there's something called the wobble where it's not always super strict. So ask your teacher about that. But anyway, so now at the top of this tRNA is something called the amino acid, in this case, serine. So this is how the tRNA arrives carrying the correct amino acid. So here, if we look at our codon, AUG, and we use the genetic code table, we find that the amino acid should be methionine or MET. So here, this tRNA that is arriving has the amino acid MET at the top. Now, if we look at the anticodon is UAC. Now, you want to be very careful here. If you use the table and you go UAC, you're going to get a totally different amino acid. You're going to get tyrosine. So you want to make sure you use the codon in this genetic code table to find the correct amino acid. So now, here's our next codon, and you can see the next arriving tRNA. The codon and the anticodon match in base pair. Now, the purpose of the codon to the anticodon is to stabilize it long enough so a peptide bond can form by dehydration synthesis between the 2 amino acids. This, the tRNA that was holding MET is now like released and it's empty. And a different enzyme will come and attach a new methionine amino acid to the top of it. Now the ribosome is going to move down, and now an arriving tRNA carrying the next amino acid serine will arrive. A peptide bond will form, that tRNA will leave, and it keeps going down. So as you can tell, hopefully, the role of the tRNA is to bring like arriving amino acids, and how it stabilizes within the ribosome long enough to form a peptide bond is by the anticodon and the codon base pairing. All right. Now, eventually, I should point out, we eventually get to a stop codon. And if you look at the genetic code table, a stop codon does not code for any amino acid. So when we get there, the whole thing signals like stop. And here we have a cute little 4 amino acid polypeptide. Normally, the average polypeptide in a human is about 400 amino acids. All right, great job.